based on a suggestion given to us when the site visit committee here was visiting. When they talked to the students, they felt there was a lot. The work is somehow being siloed, even among the experimental groups or between the experimental and the computational people. That the experimental people didn't know what data was needed for the cell simulations, and then the people doing the cell simulations didn't make it clear what data they needed and how they were using it. So they suggested that we have a student talk before the seminars, and they explained some aspect of that. So we're going to open up with a short presentation from Andrew Maytin, and he's going to talk about some experiments that we used to parameterize the SMC loop extrusion of the chromosome. Now, Andrew's only been working on this since about, what, May, April? No, a little bit before May, because since then, he's also prepared something for the advanced computational workshop in May, a very successful summer school component, but still, he's not a master of everything. And many of the things that he'll bring up, I'll address again in my talk, because we would really like suggestions, and maybe if somebody else could do these experiments besides Kees Dekker, who, nice guy that he is, he's in Delft in the Netherlands. Okay, so Andrew, please take it away.
Okay, so thank you. So like Zan said, in the theme of today's seminar, like in connecting experiments with simulation, I'm going to be talking about the loop extrusion, which is a process that's happening in the minimal cell. So just a quick introduction. So what are SMCs? So these are proteins in the minimal cell. SMC stands for structural maintenance of chromosomes. And this is a very highly conserved protein across all domains of life. So it's found in all organisms from human cells to bacterial cells. And it's present in our minimal cells. So when you try to take this out of the minimal cell dies. So it's one of the few DNA-associated proteins that are remaining in Syn3A. And I have a figure here which shows the basic architecture for all different types of SMCs. So you have these two coiled coil heads attached to the hinge. And then there's kind of like this clothesline glycine disordered protein attaching the two heads, and then some subunits hanging off of that clothesline. And it's responsible, we think, for chromosome segregation and organization. And I have this movie here, which shows SMC acting on a segment of DNA. And you can see it's kind of ratcheting or scrunching the DNA and forming this loop. So these SMC proteins extrude loops. Next slide. And so in a really nice experiment by Kees Dekker and others, they did a fluorescence study of these loops actually being formed. And the way they did that was by tethering lambda-phage DNA to a quartz surface and blowing buffer over that and imaging it with fluorescent imaging. And you can actually see as time progresses the formation of these loops. So like these red spots here is DNA being extruded. And then as you have flow over the DNA, you can see this looping form. And just to get a sense for the scale of this figure, this segment of DNA is around 40 to 50 kilobase pairs. And the scale bar here is 2 micrometers. So that gives you kind of a sense of scale that we're looking at here. And yeah, you can see also this cartoon depiction down here, what's going on. So this SMC, which is like this colored protein on the bottom, is extruding loops. And so we actually implement this looping process in our simulations of DNA dynamics. So we have our beads on a chain polymer model for a minimal cell. And the way that we kind of emulate the SMC loop extrusion process is we introduce a harmonic bond between two beads. We call them the anchor and hinge. So like in this figure on the left, anchor is black and hinge is white. And you can see as we move the white bead and act with this harmonic bond, then these kind of loops are being formed. So like this red portion of the DNA is being extruded. In theory, actually, you can have anchor and hinge pairs being formed in between different strands of DNA. In our current simulations, we just have them being formed within a single strand. So we're not going to have any pairs being formed between like this strand and this strand, for instance.
Wait, sorry, you said the single strand? So this happened at single strand level? It's not a.
Maybe I misspoke. I mean like one strand of double stranded.
Oh,
Yes.
so what sets do we know what sets the length scale of what gets extruded, sorry?
So I guess there's a bunch of different working models, but we see that the anchor and hinge pairs are only formed at the scale of the size of the protein itself. So the protein is like 50 nanometers. If you go back and like see this structure, this is like 50 nanometers lengthwise. And so you're going to only have interactions between DNA, which are at most 50 nanometers apart from one another. And you kind of saw it in this video, but this like O shape contracts and forms a B shape, iteratively pulls DNA towards itself.
But in the case decker data you showed like maybe 40K or something, I think you said, or something like that.
Yeah, so this is like multiple loop extrusions happening.
Oh.
The time scale here is on the order of seconds, so this is around magnitude of 100 loops successively. And so we can actually parameterize our simulations using experimental data. So like in the Decker experiments, they found the force-dependent extrusion rate. So if you look at, for example, like 0.2 piconewtons of force, the rate of extrusion is around 0.5 kilobase pairs per second. This force actually comes from looking at the length of the DNA on either side, and then they know the force extension curve of the DNA. So by looking at the length of the DNA, they can figure out the force enacted on the extruded loop. So that's one parameter. Another one is the actual step size. So how many base pairs get extruded every time this cycle happens. And that was done by Ryu et al. They used magnetic tweezers, and they found at around 0.2 piconewtons of force, 200 base pairs. That's the step size. Mm-hmm. So then you can do the math and you can figure out that, well, actually there's like 2.5 looping steps done per second, which is the same thing as 0.4 seconds per step. So in our simulations, we do 10 loops every 4 seconds. That's how we have parameterized this. And so I guess finally, I just wanted to show kind of all the different types of experiments that we incorporate into our chromosome dynamics. So there's like the magnetic tweezers and the fluorescence experiments that I showed before. There's our simulation model that we came up with, so like the anchor and hinge pairs. Then there's other experiments such as chip-seq, or more recently cut and run. The purpose of both of those are to determine DNA protein interactions. So in this case, it would be determining the binding sites of SMC to DNA. So where actually does SMC bind to DNA? Are there specific sequences that SMC preferentially binds to? And then finally, there's contact maps that you can get from 3C or high C data. So that can show you the contacts between different regions of the DNA. And we can actually compare the experimental high C maps to the equivalent maps that we create from our simulations. So yeah, that's my overview of integrating simulations and experiment of SNC looping.
So if I understand what there's none yet is just imaging that in the cell, is that right?
I'm sorry?
An actual image, microscope image of this in vivo, there are none?
Not that I've seen.
So in the cell, what's the typical, I guess, extrusion, I don't know if you're using the right word, that you expect? 40 kilobits, like in the in vitro, that was just a happenstance of the time point or something.
The 40 kilobase pair was just the length of the double tethered DNA that they happen to use. I guess to really know the length of these loops in the DNA, or like in vivo, that's where the contact maps would come in.
So what do they suggest to us or tell us?
Well, actually, in the minimal cell, we don't
Contact
see-
map, it doesn't unfortunately look like that. It doesn't have any major features. That was done by Ramos Dame. And so in fact, it looks like it has no persistent supercoiling. Because, you know, people started off interpreting the contact maps in terms of supercoiling or, you know, like domains. So that's hard to do. So that is one of the questions we would like to have answered. We just randomly have it interact with the DNA, we're not sure where. And the question is, we have in the cell about 200 of those, I think it's 200 monomers or about 100 dimers. But the probability that all of them are acting at any one time is low. So it is a parameter that we have to adjust. And before Andrew came along, the DNA simulations were the rate determining step for us. And he was able to speed it up by a factor of 10 to 15 by moving that alone to the GPU. So now we have the whole cell simulation done on the GPU. And then we can think about now addressing these questions because it can be done. In the summer school, they were replicating an entire genome, all 500,000 base pairs in about what, 4, 5, 6 hours? How long did it take them? So that's doable.
And it seemed like the scale is kind of experimentally kind of inflexible in terms of looking at those elements in the under the mid.
I mean, we just thought that this is the setup that Decker used was 40, 50 kilobases long. So that's about snippets of 1/10 of the minimal cell. So could, if all else fails, I will just send him sections of it. It would be nice if one of the sections would cover the origin as well. So one can't think linear genomes, but I think we do see it as critical. It affects how much bunching that you get. And that you can measure, you have something, when you have it out of the cell, you can look at the radius of gyration. It's just inside of the cell. And so this probably can even be easily, not easily, but it would be nice to have it done on E. coli.
But even in the minimal, I mean, yeah, I think Minflex seems perfect for that. Now that we, as you pointed last week, we know how to label segments of the genome. Then I think it's kind of perfect for trying to.
Well, you know how to do that. Yeah. Okay, so this is just one story. Well, nice job, Andrew. And what I want to do, and it's impossible when you do a whole cell simulation, to hit onto all the things. But what I tried to do was to give you an idea of things that have been critical and where we would love to have some additional experiments. I thought that would be helpful. And then along the line, show you how we use the experiments, whether it's wet lab or molecular dynamics simulation on a module, how that can enter into our simulations. Now, I give you, I think it's almost complete, of all the people that have worked on this minimal cell, either experimentally or computationally. And you'll see U of I has a lot of people, Take and his postdoc are helping from the Harvard Medical School. There's the synthetic biology group out at, in California. Elizabeth Vila has helped with the tomograms for the minimal cell. NVIDIA, John Stone, he's a guru for GPU computing and visualization. So being able to see your answer is also extremely important. And then from the Netherlands, we've already mentioned Kees Dekker. He's given me a lot of advice. And then Siebert Jan Marek and Eric Lindau have worked together with this Martini Gromax simulation. So we have been able to put everything together in an atomistic picture of it. But you know, you can't, at that level, you can't run it for very long. So we're just studying if we're getting the interactions correctly. And then from Germany, Julie Mohammed is going to be providing us with like 600 tomograms for yeast. And James Sense was just here in August as part of the summer school to show people how to do lipidomics on the minimal cell. And they were able to reproduce the growth rate that we also see in the calculations. But they could also tell you what lipids are available and how many of them. Okay, so again, just to introduce the minimal cell, it's about a half a micron across, and a lot have been published on it. Just to give you a feel, if you work on E. coli or Calypbacter, it's about 1/10 of E. coli in every dimension you can think of. Genes. The only thing that's really different is when you try to break down all those genes coding for proteins into these various categories, that unclear part is getting smaller and smaller for the minimal cell. Structurally, we have everything, at least a suggestion for it, in part because of AlphaFold 2 and now 3, which allows you to do bigger complexes. But in E. coli, that's 1,700 of the 5,000 genes. That's a lot. You know, and I don't like dealing with hidden variables if I can avoid it in the work. And then the pictures of everybody who has worked on this system. As I said, Ramus Dame did the 3C maps and there was no structure. And then he even did multiple restriction enzymes and we still get no substructure. So no persistent supercoiling. And then the papers that are written or listed there is where we went in and addressed various subsystems of the minimal cell, like the genetic information processing, metabolism, and then maybe the AlphaFold 2 and 3 predictions. And then we put it all together in the Cell 2022 paper, which had the first sort of 4D whole cell models presented, but it only did it for a very short period of the cell cycle, so we could ignore motion of the ribosomes, which we got from the tomograms, and we could ignore the replication of the DNA. That's our goal, is now have everything moving in all the parts, everything coupled together there. The cell was first made in a science paper. It's actually fairly famous from the JCVI folks, where Clyde Hutchinson and Hamilton Smith were the lead authors on that. And we really have to thank them, particularly as we started our work, all the advice we got from Hamilton Smith. You know, he studied mathematics here at the U of I, went off to California and decided he wasn't going to get the field prize in mathematics. So he switched into biology and then shared the Nobel Prize for restriction enzymes. So meeting with him was wonderful. And he loved listening to the theory, by the way. All right, so let me go on and before I go to the next slide, tell you this cell has a dramatically reduced metabolism. Genetic information has stayed pretty much the same as in other bacteria, but this means it has to bring in a lot of components. It has to bring in the nucleosides, fatty acids, amino acids. They do not have the metabolic pathway for those. And so you can control the media, and thank goodness, Angad Mehta really knows how to do that well. So we're able to grow it here now and have precise control over the growth media, so we can make, hopefully in the future, more quantitative investigations of how these things are affected, the various components. So, here's just a brief summary of everything you need to do a 4D host cell model. And our goal, and I hope one day I can give a talk on that, was can we, with this minimal cell, with so few genes, if we have everything included in there, can we come at the minimal rules of life? We're not there yet, but what I am going to show you was our first attempts to go over the first 110 minutes of it. So to do it, it starts with having to know, what does the cell look like? What's its composition? So that just cries out for omics. And I tell you, I have really become impressed with the Carver biotech facilities down at the other end of campus. They are really helping us out, not only for the transcriptomics, but we tried to use some DNA sequencing to validate some of the theta structures we had. We found out when they had been done out at JCVI, there had become some contamination. And even though they're in the same building, there got to be a lot of finger pointing, like your sample was contaminated. No, your multiplexing was wrong or old fashioned. The guys here, they met with us, fantastic. And they're doing it now. It's just started again this week. So you have to know omics, you have cryo-electron tomography to give you the structure. You have DNA maps, which tells us no supercoiling. Then, when you start putting in all the metabolic reactions or even the genetic information processing reactions, you do have to know rate constants or have a good way to estimate them. And also diffusion, if we're going to have this thing really diffuse through the cell. Again, we're lucky there have, we've learned very much how to how should I say, approximate these things from other experiments that have been done. And there's no other way around it because so few experiments have been done actually on this minimal cell. When you're propagating the state of the cell in time and in space, you have to involve hybrid methods. Some are going to be stochastic. I think all of us agree you can't say gene expression without saying stochastic gene expression. But when you have more higher particle counts, then you can go over and use ordinary differential equations such that in treating, say, the many of the metabolic reactions. So that's what that little picture is trying to show you there. So we have coupled to those reaction diffusion operators, we have ordinary differential equations for metabolism. And now that we've implemented the DNA model from Andrew, that's yet another methodology coming in through Brownian dynamics. Now, all this has to be run on the GPU. If you want to try and get these things done in a timely fashion, and timely fashion, as we said at the beginning of Andrew's talk, is such fast enough that we can try out various parameters to see if we understand the SMC looping, the role of the topoisomerases, the two essential proteins that they could not knock out in the cell. How do they interplay with one another to have this large scale motions of the DNA? Everything goes well. You can then come up with the messenger counts as a function of time. Look at the variation in doubling time. This cell has so little regulation, there is going to be variation. And the same way we've been able to calculate as we go along the ATP costs for everything that we've included in the model. So we already, I think I showed this one time here, that this is from the 2022 paper, when we only did a very short time, 20 minutes out of the 110-minute cell cycle. So we're not going to we won't have DNA replication. Those are in those orange beads. The yellow are the ribosomes we got from the tomogram. They're also not going to move. So what you're going to see here is the fate of a messenger once it gets, expressed, say from transcribing RNAP, and then the ribosomes will change color if they're translating a ribosome or not. And then the red dots are degradasomes. That's a complex that will chew up the messenger. And again, they will change color when the messenger is present there. So if you just watch this and you do it again and again for many, many different configurations of the DNA, what we learned from this was approximately the half-life of all the protein coding genes. But we also learned how many of the ribosomes are typically active and how many of the degradasomes are active. And knowing that number, we could come up, we projected how things would go for the rest of the cell cycle by coming up with probabilities of binding to degradasomes or to ribosomes. So much of what I'll show you at the beginning, we did 4D, but only at the beginning and not even what I'd say complete 4D, but to learn, help us get the parameters to be able to do the longer simulations. And by longer, I mean, if we have this mixed stochastic diffusion methods, we talk of a reaction diffusion master equation, like on that top line there. And then not even showing you the ordinary differential equations for metabolism or the motions for the DNA. I'm just showing you the reaction diffusion master equation. So the red part is just over the reactions, and the bottom part is over the diffusion. And you notice there's another subscript there because we take the cell and discretize it into subvolumes. And based on that, we can have maybe something moves out because you or can't get into that subvolume because a ribosome is there, or a protein interacts with the DNA in a step. So things can diffuse between subvolumes, or they can react within the subvolume. And again, how you break up the reactions, a lot of it depends on time scales, abundances, and the differences in the kinetic parameters. Now, whenever you have mixed methodologies, you have to know when do they need to communicate with one another. So we wrote a paper back in 2018 in one of those IEEE journals where we could show, how often or how infrequent could we communicate and still not create a large error in the simulations. And not only do you have to think about the communication times, but also what gets communicated. So maybe the enzyme counts out of genetic information processing. In metabolism, it's the NTPs that get made, but then you have to assemble it into a mRNA or the deoxys into the DNA, and then the recycling from the messenger through the degradation by the degradasomes. So it is even more complicated in the cell paper. So I don't want to put up that diagram, but you get the idea. And so just looking at metabolism, I always make this joke, you can write it on a single page, you can't read it, but you can write it. And every student then took a subsystem and became the expert on it, read the articles, knew the kinetic parameters, we made models, and then we'd get together and see if they were compatible with what we knew about it. And so there were, if we're going to do a full kinetic model, we needed a lot of parameters, but at the end of the day, you have to do some machine learning and free energy analysis of the final choice of kinetic parameters you take. So for example, let's just look at that center, the central metabolism where the sugars come in, and that will then be a connection over to lipid metabolism as well as over to nucleotide metabolism. And it has the main fluxes in just about any cell. To begin with, it is so important because unlike other bacteria that you may work on, this has no oxidative phosphorylation. You only get ATP from that central glycolysis pathway. And then on top of it, nobody teaches what happens when you have more than one substrate binding and giving you more than one product. That's called a random binding model. If you know nothing about the order in which they come, you're happy if you have some KMs for those, which then could go in for an expression for the rate of that reaction. So the first term is like the forward reaction, and the second one is the backward reaction. And that's the most general equation that you can use. Now, where do you get those parameters? Because as I said, so little has been measured for the minimal cell. Well, we have to thank our biochemistry friends for decades of measuring kinetics of those individual enzymes. And they have, to a large part, been deposited in Brenda. Of course, different temperatures, different species. So you have to use these Bayesian estimators to come up at least with initial estimates for these parameters. And then if you start tinkering with the forward and the backwards rates of these things, you better check that you get the right thermodynamics drop in energy as you go down that pathway. So if this would take you back to your P-Chem class, where then you would, hey, oh, how do I calculate a free energy from like energies of formation? If I knew the chemical potentials, I gotta get delta G0 first, and then I look at what the concentrations are at some point. And so you're seeing up there at the very top, the delta G, and those are just the enzymes that are along that pathway. Glycolysis pathway. And you have several different ways which you can calculate delta G0. There's even a program that's pretty good that's on the internet. It's called Equilibrator. I think Albert's sort of one of the leaders in biochemistry and also in life sciences put that article. We can look at how the cumulative effect of the delta G drop. And then last but not least, you can look at the fluxes. So at each time, sort of major time intervals of the cell, you can see whether is it reaching what you would call steady state. And sometimes it's above, sometimes it's below, and sometimes it's near what would call the steady state solution. But you see, we don't always agree, even using two different methods, like just from the kinetics. Most of the time, the crosses and the X's are pretty close. But for FBA, that is a problem. There's also enormous disagreement in the Brenda database for that. But you know, you do the best you can. We've tried several things. We found a set that seemed to give us the results that were consistent with a number of global experiments. And one of the most important things for us was getting at the rate of formation of the lipids. So let's call it lipid metabolism. So this cell brings in fatty acids. And for those of you not used to looking at pathways, at the top, it has like fatty acid or FA_E, that just means it's outside of the cell or extracellular. Then it's brought into the cell, then it's _C. But if you go down that pathway, you'll see that at first it makes a single a lipid called PG, and then you put two of them together to get like a cardiolipid. Now, those are the only two the cell actually makes. But this organism, it was derived from a Mycoplasma mycoides. It is able to take up lipids from a host. So we can take up sphingomyelin, that's SM or PC. And thanks to James Sense, we were able to know what is the fraction of each one of those in the membrane using the growth media, which contains all those in an experiment that was done by Kim Wise at the JCBI. All right, so when we held the workshop, and as I said, led by James and his assistant, who came, she's from actually North Vietnam. I asked her why she didn't come to an American school. She goes, Zan, the war? And I go, oh my God, they still haven't got over that yet. So it's easier for them to apply to Germany, where they feel, but she was very happy here. So I think she's very young, but she's finally forgiven us, right? Anyhow, what I'm showing you in this picture is the Martini model of those lipids. And you will see like cholesterol, there's a lot of it in this cell wall. The feces are purple, there's PG and the SMs are yellow and the cardiolipins are brown. And this was only possible, we could get an idea about them when we were able then to make this whole cell Martini model. Now, we do not know we only know composition. We don't know how they're grouped together in the cell. That's one of the things we'd love to figure out, if somebody had a metric for cardiolipins, to know, are they displaced all over the cell membrane, or are they located in certain patches? And again, I cannot say enough and thank Angad Mehta and his lab, because they also provide the media for James, because what got ordered was not the right one. So I think he exhausted his entire supply for the summer school, and also helped grow the cells, had them in the freezer so the kids could just warm them up for the summer school. Now, here's what they did in the summer school. We borrowed a shaker from physics. Thank you, guys. And then they grew it with 3 different conditions. And then we're able then to isolate the lipids, do a thin layer chromatography, and get at the amount of each of those components that I just showed you before. This is just one of the many experiments they did, but here's the nicest one. They did a growth curve and got a growth of about 99 minutes. Now, what JCVI had measured and what was in our cell paper was about 105 minutes, but they had done it by labeling with pico green. What they did here in the summer school, they used optical density at that particular wavelength to do it, right? So what else goes into this membrane are membrane proteins. And here we relied on some experiments and crystal structures that told us what these translocase complexes should look like. There's a sec Y, sec A, they come together. But then the molecular dynamics simulation of Gumbart at Georgia Tech was able to give us the speed at which how many amino acids are threaded through and then at what rate. So that all went into our simulations. So when we simulate the growth and the surface area of the lipid, these are the numbers that we are using. And that's how we got these curves. So it was from that metabolic lipid metabolic model that I showed you earlier. And then we noticed that the volume doubled and the shading just indicates, like we've done on several replicates, that we had about 50 to 70 minutes. It took that cells on average to double its volume. And then the rest is sort of an assumption we made that it more or less undergoes symmetric cell division. But we can also look at over on the right-hand side, just the change in surface area from the original to its final one at about 100 minutes. We could compare in our simulations how long that took, compare it to the experiment. And then to get the surface area, you have to allow for the contributions from those many membrane proteins that I showed you at the very beginning, as well as the lipids. And again, you just do standard area assessments and putting those in. So it looked pretty good, and that's why we were quite happy to run with this model. But as I said, there are aspects of it we'd love to have checked. But as I said, nobody can tell us where cardiolipins are in any organism at the moment. But the fact that it was symmetric, well, lucky us, because Angad had grown the cells here and had helped with the labeling of not only the DNA, but the membrane, but also a protein that's involved in the septum. We could take his postdoc, Jiwon, come in and then do measurements sort of at an early stage and then at a later stage of cell division. And I think in the top, particularly in that third panel, you can see very nicely in the early prolates state, the DNA there seems to be a lot less density in the midplane. The FTSZ is accumulating there. And then down here, it looks much more symmetric. Now, if you get out and count the pixels, it's not really quite symmetric, but hey, we're going to assume it's symmetric and impose that because we do not have at the moment a good model for how the FTSC septum is formed, because it's just not FTSC. There's an entire opulon of about 6 proteins that are involved in it. And while one's learning more and more all the time, we still don't know exactly how they interact and how it polymerizes to form the septum. But those measurements were done here. Sent Jiwang in here to the U of I, and they went down to IGB and used one of their Zeiss machines, and were able then to do these measurements. Now, that was just sort of talking about the membrane and metabolism. I'll go as quickly as I can through the DNA replication, since Andrew gave this nice introduction to it. We do know that replication starts with the binding of a multi-domain protein called DNA-A. And I know that Paula Mayer is very aware of this protein and has been studying it in her work on Calypbacter. it binds to both double-stranded, one of its domains to double-stranded DNA as well as single-stranded. And here are 3 signatures, each 9 nucleotides long, where the one domain binds to the double-stranded DNA. So we think what happens is it twists it, opens it up, so then the other domain of DNA A can then bind to the single-stranded DNA. So down at this picture, whoop, that's the pointer. So it opens it up and then you start getting a filament formed. And when we discussed this with Hamilton Smith before, this is the only thing where I've noticed he may have made a mistake. These are all, what's underlined are pretty good signatures, but later one of the students noticed, hey, in the middle of this thing, there seems to be a CCCC. Oh boy, that's not good. So actually, you can't really bind 30 in these organisms. You can only bind about 15. And when you put that into the model, we get a much better agreement with the ratio of the origins to termini. But again, what will be the final validation of that will be the DNA sequencing. But the kinetic model is unaffected by whether you have 15 or 30. Where did we get the parameters from? Again, there was a wonderful FRET experiment done where you flowed the DNA A through and they can measure the on and the off rates. We used those. We didn't take the first step. We took it when they were elongating it from that paper. And then for the polymerization model, initially for the just doing the well-stirred model, we had a polymerization model where all you had to identify was what is the template? Is it single-stranded DNA or messenger? What's the protein that is helping to make the polymer? And what are the monomers used? Is it NTP or the deoxynucleotides, or is it the amino acids? So this model had been published by a physicist, Hofmeier, already back in 2013. Zane was able to modify it, Zane Thornburg, so that we could use it with our present knowledge. And then we could then at least go through one phase of replication. And in some cases, we got multiple forks being formed. And here I'm showing you if the blue balls are the origins, and the yellow squares are supposed to be the terminus, and you look at the various ratios you can have. So you can also just have duplication or close. You start off with 2 to 1, and then you can get 2 of them already made. But even after you form 2 of them, you can have multiple forks in either one of them. If you go over and just look at the origin to ter ratio in the exponential phase, which is the far the blue to the far right, you see they were getting a value around 3.2 or to almost 3.5. Some of the simulations were getting close, but huge error bar because we couldn't do that many. So now we're really trying to understand that. And first you have to come up with a way to classify the theta structures. So this was the work of Ben Gilbert. So he was looking as the cells are making the daughters, the left and the right. And then each one of those can start forking. And then when you undergo cell division, it could be that there's nothing in one of the cells, but it could also be that it gets split. So one of those structures goes into the one cell, and the other one goes to the other one, and then you follow each of those. And you keep doing this again and again and again for 100 cell cycles. And if you, on the far right, look at the histogram at each time point over the cell cycles, using the present model we have, you get an answer that's about 3.8. And if you look at the time-dependent nucleotide counts at each position along the genome, and 0 would be the origin here, you get this plot. And then if you take roughly, that's about, say, 11, and down here it's say 3, so then 3 into 11, yeah, you're getting close to 3.8. So that's roughly how you get it. It's not exact because it's depending on how you do the averages. But as I said, we need that done experimentally to validate the model. And the experiments are being done, as I said, here at the Carver Biotech and using the Illumina sequencing. No matter what stage of the model that we're at, we insist on then running it and then see, one of our tests if the model is performing at all. Do you double the counts of proteins over the cell cycle? And do you look at the time-dependent behavior of everything that's in the model? And maybe just a last comment or two. One of the things that can happen, and again, it just sort of, you have to be careful about what is considered, oh, everybody knows this type of thing. If you just look at nucleotide metabolism, and you can see that you can only bring in this diagram, bases or the nucleosides. And in the medium that they were growing, they only had the nucleosides. But in about 2 or 3 steps, unlike E. coli, which takes 40 to make it the whole thing, the nucleotide, this is done from a nucleoside in about 3, and you get GTP. And you better get enough to make the messengers, and the same thing goes to be able to make the DNA. So you also have deoxynucleosides in the medium. You have recycling from the degradosome, and then you look at what the demands are to get the messengers, to make the proteins, and here to replicate the DNA and satisfy those other more global experiments. And so most people assume that the pool size stays static, not with this minimal cell, which really lives on the edge of life. Those are the rates of the polymerization for transcription, translation, and DNA replication. And occasionally, if you run out of monomers, which is down in the denominator, that's 1 over something 0. Well, that's not good. And so it nosedives, right? Now, most of the time, the cell will correct. This was just for a particular gene that had happened. The black line, or the average, or what we call the healthy cells, but occasionally some of them can die, and we try to dynamically represent how the central and nucleotide metabolism are connected in order to help make these NTPs and the deoxy versions of them. So over on the far right, we have the glycolysis, the metabolic pathway for that. And then we have some of the genetic information processing steps over here. But what happens, we've noticed, this can just start collapsing somewhere involving these enzymes, and you'll run out of PEP, and then everything just starts dying when you get cells out of maybe 200 runs. At the time for this, for our paper and cell, we were getting about 80% of them were surviving, 20% would die. And this is what I showed you so far, mostly the results from doing a well-stirred model, but then to move fully into a 4D over the whole 110 models, we had to change the DNA model from doing a lattice polymer picture to one that had like beads on a string. I don't, Andrew didn't show this, but it has the normal bending and stretching and excluded volume, so you don't cross the DNA strands. You have self-avoiding with the ribosomes, and then also there are beads that represent the interior of the surface of the cell, and it can avoid that too. That was done in 2023, and with that, getting a set volume, we calculated what a state would look like. Gave it off to Marek and Eric Lindau and Siebert Jan's student Jan, and they made this all atom model. Once we also provided them with the structures. Now, we had a model that was a perspective, perspective in Frontiers of Chemistry, but we couldn't simulate it until Eric changed GROMACS to be able to do anywhere from 500 million to 2 billion particles. But at the May 6th Advanced Computational Workshop, they were able to do that. And here is the first 100 nanoseconds of this a mini version of the minimal cell. And all the students were able to construct this and simulate it. And then we also brought in our colleagues, Shulei Wang and his student, Rong Wei, to look at the cells that did die and could we see anything exceptional about them. Because up to the moment they died, everything looked pretty much the same as the healthy cells. And so we're beginning to unravel that. And now I also want to show you this because this is ongoing work with the all atom model. Again, this is being validation, but from a computational perspective, but another method is they've also put in the metabolites. And now we can zoom in and just look at how both the proteins as well as the metabolites interact with the DNA. And now I won't bother to show you this, I'll just start it. Of course, everything we do gets put into Minecraft. And I really thought this would be cool because now look at the resolution. We had at 0.4 nanometers. You see the red are proteins that interact with the DNA or in the cytoplasm, actually. That's the DNA. You could walk along it. And then the blue are the membrane proteins. And of course, the green are the membrane. But more importantly, what Andrew was showing you was what happens when these two essential proteins, which they could not remove, what are they supposed to be doing? This is the common wisdom, is that the topoisomerase makes these loops and it can do it on either strand, but sometimes the strands get entangled and then you have to have the topoisomerases to break those. And that's exactly what we do in the simulation. So that picture you saw, and then this is what one was able to calculate. And the yellow is the new DNA that's coming out of this. And you can, the red will be the origins of the two chromosomes that are being, the old one, new one, and the anchor and hinge are black, which is not a great color. And but you can see the hinge positions here and they jump around. We would love to know, get a better idea of really where they bind and more details about it. But we're hoping this is good enough to be put into our 4D simulations. So if we don't have a kinetic model for the septum formation, what we had to do is rely on yet another energy function, which would try to minimize the surface of the cell using the information we generate on the surface area and the volume. This is called a Helfrich Hamiltonian. And we're working together with Pozeskian, Viria Pozeskian, who's developed this code. And we've applied it. This is on a simple model already. It's almost half a year old. And you watch, and the volume's already doubled, but the transition to that other prolate state is a little bit too sharp. So we've been working to improve that. But the main problem that we had was the DNA simulations were just so slow until just recently. We could not add to it the DNA. I'll show you an early result we had where we tried this. So same thing, ribosomes are changing color when they're being occupied. Now we put for blue the, I think that's the sugar transporter. But what you see is even though the origins are opposite side, there's just a lump of the DNA in the middle. It hasn't spread out enough. But now with this new version and the of it, and we found there was an error in the minimization, we're able to have it spread out. And I don't know if Zane has finished the calculation. He just got the data from the program from Andrew, started using it this week. And we would hope then that this is now, this story is almost coming to an end. And so just to summarize quickly, what I was trying to show you is how we go from using experimental information and how that enters into our computational. So sometimes, we look at you guys like this, and I know that you think we just do this, right? But it's important that it's not one way, but it's two way, and that through our computational models, we get back to the experimentalists and then try to validate, what we're using, and this hopefully get at those minimal rules of life. Now, this was just a story about the minimal cell. We've also started to do this on yeast because so much has been known about it. And again, we're very lucky that Angad Mehta is one of the experts on this campus. And I cannot emphasize enough how important it is just to be able to control the microbiology of these systems and be sure things don't become contaminated so we can't get a straight answer. and when we try to either change the medium or, want to get additional information from it. And for those of you who are interested in working on yeast, we'll probably start up a little, thrust, with ONGOD students in mind that are working on yeast, so we can help design the experiments a bit better. But with that, I hopefully have given you guys an idea of how necessary experiments are for our theory and simulations, and I hope that people can learn from our simulations. And as I said, I want to also thank Andrew for doing such a nice job of explaining what he did. All right. We'll be happy to answer any more questions, if there are any. Hi, Paula.
Hey, thanks. That's cool. I'm wondering about this death, the cells dying.
Uh-huh.
What? Because you showed that it was nucleotide, maybe they started running out of nucleotides.
Well, one thing I didn't emphasize is two of the proteins that are in the glycolysis pathway that convert ADP to ATP, they convert any diphosphate to any triphosphate. That's PYK is there, but the main fluxes are in the the glycolysis pathway. So if you start having problems there, then you're running out of either PEP or ATP, and then things just start crashing. So one of the things we'd like to do is be able to dynamically represent those connections so you could see, but the machine learning people at least have been able to take the trajectories of the healthy and the unhealthy cells and tell us and give us a little bit of, they can identify the ones that are gonna die earlier than what we did with our simulation.
Because I'm wondering if you can tell between just the cell stops growing because they're lacking the necessary nutrients from actually dying.
That's when we saw them dying, and we've defined death computationally, right? We asked our collaborators who are doing the experiments, and this is JCVI, how do you tell if the cell is dead? And they're looking at a population and because they just measure picogreen, that's a metric back to the DNA. So it's not looking and telling you you don't have anything at a single cell level. That would be great if one could do that. So first we want to look what maybe the machine learning is telling us, and then go and do even more of that, to see if we can't cause a bigger section of them to die, so that we can really say, yes, it is these couple factors, right? But it's never going to be it's like cancer. It's never going to be one factor that causes things to go south. There could be multiple ones.
I guess what I'm confused is how does the computational, how do you know they're dead? How do you translate death from the computational?
Well, you look at the levels of PEP and you look at the levels of ATP and you start running out of them.
And that's how you define
Yes.
them. Okay.
Right? And as I said, that's why I showed those transcription, translation, and replication curves, the rates of those elongation, because sometimes they dip down, but the cell can recover. Right? And it might take a little bit. And remember, this cell has so little regulation. This is not going to happen in E. coli, and it's not going to happen in Calypbacter. Right? So regulation is extremely important of keeping things within the range that you need. But that's also why I like the minimal cell, because it's just putting in those features and seeing what happens, right? Okay, any other questions or comments? So, yeah,
I have
nice.
a question about the surface area that you calculate from the membrane story. So how did you calculate the surface area by the membrane proteins? Did you basically
Well, we know how many membrane proteins are coming in or being made and then being inserted from our calculations. Now, this is sort of something that people who do molecular dynamics simulations of membrane proteins do a lot. You can come up with an estimate of the surface area that membrane protein takes. So we took an average of those, and the same way for the lipids. Could we improve on that? Yes, but no need. There's a range that's even been assessed for that. So that's how we do it.
I see.
So we assign for every lipid a certain surface area, for every membrane protein a certain surface area, And in that plot, there were two sets of curves. One was just for the proteins, one was for the lipids, and then we put them together. But like, this thing is full of membrane proteins.
Yeah, but it's 50%,
Yeah.
right? So is it like underestimation, do you think, or is it?
So far, because we came out with approximately the right doubling time, it's not pressing to look at it. Could you have improved on this? A bit. I prefer to wait until we can label a few of those things. So like even the membrane proteins to bring in the sugars. Can you tell me if they're all clustered together? We saw some preliminary studies from Take that they, or multiple clusters of them, not just one, but it's just based on the intensity. The intensity still was not able to give us how many are there, but we have 1,000 of those PTSGs. And I don't remember how many of the nucleoside transporters and the fatty acid transporters. So there's a lot to determine, and it would be great if we even know where they're localized. And some of these are questions that the people who do MD on membrane proteins are just beginning to answer. Like when I talk to Ahmad, they're seeing more and more how cardiolipids interact with membrane proteins is much different than some of the other lipids do.
Quick question.
Yeah.
So, when we talk about the topoisomerases and like how it's implemented.
We just weaken the potentials and allow them pass through.
Okay, but that topoisomerase action, are there like several conditions that has to be met? Like, is there a specific tension?
No, I guess, don't you wish? When